to put some numbers around that just for grins, like 1% of the genome is the exome, is this code
Correct,
part.
correct,
And
correct. So
and I've
yeah,
seen that the estimate is about 85% of the clinically significant mutations occur in that. So
correct.
the other 15% occur across the other 99% of the genome. So it begs the question, are you better off sequencing the entire 100% of the genome and getting 100% of the clinically significant mutations? Or are you better off sequencing 1% of the genome and capturing 85%?
Welcome to the Precision Medicine Made Simple podcast, where we simplify the complex world of precision medicine. We're here to explore how personalized healthcare can empower you to understand and improve your health to live a longer and healthier life. Join us as we break down the latest in medical science into actionable insights. Let's make precision medicine accessible and actionable for everyone. Enjoy the show. Today's episode is sponsored by Biography Health, where healthcare meets personalization. With their DNA-enabled blueprint, Biography Health empowers you with a health plan based on your unique DNA, aiming to prevent disease and extend your life. Take control of your health journey with a plan as unique as you. Visit biographyhealth.com and start your personalized healthcare journey today. As a reminder, this episode is for educational purposes only. This episode is in no way a replacement for professional health advice. Please consult with your professional primary care provider for medical advice. If at any point you feel that a topic discussed on our podcast was misrepresented, or if you have additional insights that could enhance our discussions, we wholeheartedly encourage you to reach out to us. Your feedback is invaluable in helping us improve and in ensuring our content remains both informative and reflective of the latest developments in precision medicine. In this episode, Dr. William Stanford, Dr. Yelena Bykhovskaya, and Taylor Koo discuss various genetic testing methods. The conversation compares genotyping, exome sequencing, and whole genome sequencing, with an emphasis on whole genome sequencing, which provides the most comprehensive genetic information. Hey everyone, welcome to the Precision Medicine Made Simple podcast. We are doing a quick episode here for this week. I'm joined here with Dr. Stanford, and then last episode's guest, Dr. Yelena Bykhovskaya. Welcome back, you two. And for today's topic, we're going to be discussing the different types of genetic testing. So the differences between genotyping and the different types of sequencing that's out there. And so I am grateful to have two renowned experts here guiding me through this concept, because I have plenty of questions to ask as I've seen these terms getting touched on before in previous episodes, and then also from other companies as well. So I'm excited to dive into this conversation. So touched on this from the last episode, but something that I've noticed, and let's just say from a broad stroke of like 23andMe, right? Like the other in Ancestry.com, they always talk about genetic testing. What type of genetic testing do they do, and how is that considered? Is it genotyping that, from my understanding of how they run their genetic tests?
Yes. So that's correct. So, well, we should go back a little bit and just let's just review what is it that we can do with our genomes. So our genomes are rather large. There is 3 billion base pairs of unique nucleotides that form it. So that's a lot of information. And this information wasn't known for a very, very long time. And so what happened is that we kind of got the bits and pieces of the information even as Human Genome Project progressed. And as those bits and pieces became available, people wanted to analyze them, and they wanted to be able to do some analysis and make some decisions about genetic risks and mutations and identify disease genes. And all of that became possible by basically analyzing the parts of DNA that we kind of knew about. And that's how the genotyping kind of came along. So the genotyping panels are basically different from any sequencing platform is because they use the already known information about already known genetic variants in the genome, and they place them on this platform, tiny chip, which is called microarray. And that chip kind of is placed in the machine, and we apply DNA sample to it. DNA could come from saliva, or from blood, or from tissue, or from frozen tissue, blood spots. So there's a lot of variability in the substrate of DNA that you put, that where you get the DNA from. But at the end, you just analyze this DNA sample, and you get the information about these certain positions that are on the chip. So the first microarray chips were kind of small. They only had 100,000 base pairs, 100,000 positions. Okay, so
Positions.
Unique positions. Yeah, it's not base pairs, it's unique positions. So 100,000 unique positions. And then these positions were used by researchers to make decisions and identify some of the genetic susceptibility loss, actually, for some diseases. So the initial genome-wide association studies, which are very important studies that were done in the beginning of 2000. So the first one was actually published in 2005. They were done with this novel platform, which was genotyping chips, and they were very successful for what they were. So if a single gene or single variant was identified at the genome-wide significance level, which is a very high level of statistical significance, then it was deemed to be very successful, and so it was. Then those chips became much bigger, right? And the two companies, one is called Illumina, and one is called Thermo Fisher. They became the leaders in the field. So if there were other producers, no one remembers about them anymore, because they were all acquired and merged into one of these two big companies. So basically, every single lab that runs these kind of platforms in the US or in the world, they would use either one or the second company. So 23andMe uses Illumina, and they use one of their more advanced chips. So actually, that chip has about 400,000 variants, or they're also called SNPs, single nucleotide polymorphisms. And so all the information that they get from this chip is that and that's the information they use for all kind of analysis that they provide to their customers.
And for clarification, when we're talking about chips, then I'm assuming like this is sort of just like a sample size of the, I guess, of our DNA. That can you elaborate on what chips mean?
Yeah, what's a chip look like? What are the dimensions of a chip? If someone put a chip in front of me, would I know what it is?
Okay, well, you know what?
I think like a microchip.
The microchip? No, they're not micro. They're this big. So I don't know, like 2 centimeters by like 4 centimeters, something like that. Okay, or like 1.5 inches by 2.5 inches.
They have a lot of nucleotide sequences that are known, and they pop up the donor DNA, the patient's DNA, and the ones that stick signifies that the patient has that particular sequence.
Correct. So it's called hybridization. So you have something called probes, and those probes are either so your DNA is sheared, it's kind of cut into small pieces, and then it's being hybridized to this chip, to this product. So if you match, then it's one kind of a signal. And so if you match with two copies of the same variant, then that's one kind of signal. If you're heterozygote and you have two different nucleotides at the same position, then it will match to two different probes. And so if you're homozygote or have two identical nucleotides in that same position, then it will be another, another signal. So all of that is actually being captured, but there's a computer program, and actually the whole process of analyzing the data from 400,000 variants only takes hours to do. So the project is actually very streamlined, and the accuracy is high. And the main disadvantages, of course, is that you are only looking at what's on the chip, at those set positions and set nucleotides.
Okay, so let me see if I'm understanding this correct, because it almost sounds like the chip is almost like a cross-reference then, and seeing which part lights up within the chip, and then that is how you're able to identify whatever is on the chip, then that DNA has that sort of variant as well then, right?
Exactly. So like for those, if Dr. Bykhovskaya was saying it was 400,000 discrete locations that we're examining, say, on the chip. And again, that's out of 3 billion base pairs.
That's correct.
Genetic code. So a tiny fraction out of, I don't know, there's probably 100 million different possible points of variation, at least 100 million possible points of variation in the genome. So you're getting a small amount of very basic, valuable information. And the technique, as we've described, is chop up your DNA. If it sticks in a spot, that means that you have that gene variant. If it doesn't, that means you don't. And that information, as cursory as it is, can be very valuable and useful, especially given that the chip is designed to give that very high impact type information. Now,
correct. So everything that's on the chip, these variants, they're not random. They're very carefully selected for being important. So what are they for? So first of all, they are for identifying and calculating your polygenic risk. So the polygenic risk scores came from genome-wide association studies. And genome-wide association studies used this they're called common polymorphisms. So the ones that all of us, or most of us, carry, and they looked at frequencies of these polymorphisms in individuals with certain diseases and individuals without these diseases. And they compared the frequencies of those SNPs, of those polymorphisms, and some of them were found to be greatly associated or linked to certain diseases and certain phenotypes. And so the polygenic risk scores actually consist of multiple variants or multiple polymorphisms or SNPs that are all over the genome, located on a number of different chromosomes, and their frequencies are being calculated together to make a polygenic risk score. So that's probably the most useful function of the chip, of the polymorphisms on the chip, right? So that's number one. Number two is actually pharmacogenetic analysis can also be done using information from the gene chip. And it's because some of the variants that are known to be functional in affecting the abilities of the enzymes that are involved in drug metabolism, they're also known, and so therefore they have been preset on the chip. They're available, so we can get information for those markers as well. So the part that's the least informative on the chip is the part that involves monogenic variants, rare variants, variants that not many of us share. We all carry some rare variants, but a lot of them are very unique and rare, but this by definition. So there's millions of those variants, and it's not possible to put them all on the chip because the chip is only 400,000 base pairs filled with 400,000 base pairs of markers. SNPs, right? So that's where that information will be lacking. The other part that will be lacking is the ability to identify novel things or things that are were published recently or revealed recently. So the genetics is probably one of the most fast-moving fields, and there's new information that gets deposited in the databases every single day. So having a chip and analyzing a chip lacks the ability to be able to analyze this novel and emerging yet important variants and additional positions in the genome because they're not there yet.
Now, potentially a very dumb question here, and apologies if it is a dumb question, but are there different types of chips for different types of analysis then? So is there a chip specifically for genotyping then, another chip for exome sequencing, another chip for microarray, and then ultimately the best chip for whole genome sequencing then?
So when you say the word sequencing, that's not a chip. So the chip is for genotype, right? So genotyping is what's on the chip. So sequencing is a different process. It's when the probes are basically all over the genome. So they span the whole genome, and your DNA, which is also being cut in little pieces, right? But it's being aligned to those probes, of which there are millions, okay, and billions, I don't know, no, probably millions, okay, 100 millions that are being aligned. So the sequence becomes a continuous, it's a continuous line, basically, if you think about it, right? From the beginning of the chromosome to the end of the chromosome, right? So that's a different process. And this come as kids. So again, there's no chips, and this is now sequencing we're talking about. But you're actually onto something, because genotyping chips actually could be modified and based on the purpose of what you're trying to do. So, for example, you can take all the variants, all the SNPs that are known from whole exome sequencing, right? So let's say you sequence 10 people, okay? You identify all the variants that are different between them, and then you put those variants onto the big chip. So some chips could contain as many as 5 million of base pairs. That's what I remember. Okay, maybe they're bigger, but that's what I remember. So therefore, you would get, again, the same idea that you only look at what's on the chip. But now you have many more positions, many more SNPs to look at. And so, yes, you can modify this chip. You can customize it. For example, for pharmacogenomics, you don't want just just a handful of known SNPs. You want to add some additional ones with what's called so-called emerging evidence. And so you can do that. and you just add them to the chip and so forth. So the chip is not is a powerful way of looking at the genome, but of course it's not the most comprehensive, but because you will never be able to see everything. So that ability only comes with whole genome sequencing.
So in this, correct me if I'm wrong with this analogy, but this is sort of what it sounds like. Because as I was doing more research into this, it almost sounds like you have this book, like an entire book, right? There is, and the book is all, let's just say all of the words and chapters are all blurred out. And so with genotyping, then there's specific words that are identified then that kind of paints the picture of like the whole story of the book. And
I love that analogy. That's beautiful. So the analogy would be with genotyping, the question is, does the word Mississippi appear on the last line of page 23, yes or no? If yes, then you have the Mississippi 23 allele.
Okay.
If no, then you might have the Missouri 23 allele.
Right.
And whole genome sequencing tells you every word in the book, period, full stop. So that's kind of the fundamental difference. What a great analogy. I hope that's not copyrighted by you, because I'm going to use that one.
Yeah, please use it. And so then if whole genome sequencing is the entire book, then I'm assuming then with like exome sequencing and then microarray, it's almost like just chapters or sections of the book that paints a very particular story just for that specific chapter.
So the microarray is back to that, more discrete pieces of information. Does the word Mississippi appear, as the second to the last word on page 23? That's like the gene variant. Whole exome is also sequencing. So it basically tells you all the words in the book contained in the middle of the chapters, but it doesn't tell you what's in the first paragraph or the last paragraph or lots of paragraphs. It does sequencing of discrete areas of the book, which are called genes. So those genes, so that coding part of genes, and let's just suffice to say that a whole bunch of the gene does not ultimately code for pieces of protein that become protein. So it's a lot more information than is contained in the
of our whole genome.
And to put some numbers around that, just for grins, like 1% of the genome is the exome, is this
correct.
And
yeah.
I've seen that the estimate is about 85% of the clinically significant mutations occur in that.
Correct.
So the other 15% occur across the other 99% of the genome. So it begs the question, are you better off sequencing the entire 100% of the genome and getting 100% of the clinically significant mutations? Or are you better off sequencing 1% of the genome and capturing 85%? For my practice at Beverly Hills Institute for Precision Medicine, I think also obviously what we're doing at Biography Health, is I've chosen to do the whole genome sequence because the extra cost to me more than justifies or is compensated by getting that extra 15% of clinically significant SNPs.
Absolutely. And I agree with that. And also, this is an emerging field. A lot of labs and a lot of research institutions all are changing and switching to whole genome sequencing for that same reason, that people don't want to miss something that's important. Technical difficulties, limitations and price. When whole exome was a fraction of a price of a whole genome, then that made sense because more individuals could have been analyzed. But now the cost is no longer prohibitive, and the difference is no
They're on
longer.
the same order of magnitude.
Exactly.
It's not as if sequencing 1% of the genome is 1% of the cost of sequencing the entire genome. It's more like a factor of 2, if that.
Yes,
like
yes.
$450 to sequence 1%, and it's like $900 to sequence the whole thing. So
that's right. So the way I look at it, I think that's how we'll look here, in biograph health, is that we started with genotyping. It was good for what it did, but the technology moved on, and whole exome sequencing was also good for before. So all of this is in the past. We're in the present now and in the future. And for present and future, whole genome sequencing delivers everything that whole exome does, and the genotyping, even at the larger scale, genotyping does, plus more. And it's technically very doable and delivers a high quality sequence.
I appreciate the two of you breaking this down. It's definitely going to be an episode that I'm going to have to listen back to. And then in case anybody has questions and wants to just discuss further, what's the best way that they can contact you too?
I think emailing is great. It's my first name at biographyhealth.com. So
it'll be in the show notes. Thank you two. I appreciate your time, and thank you everyone for listening.
Thanks so much.
Thanks for joining us on the Precision Medicine Made Simple podcast. If this episode inspired you to learn more about your health, please leave us a review and share the podcast with a friend. Your feedback helps spread the word about the power of precision medicine. Remember, taking action today can lead to a healthier tomorrow. See you next time.